And good afternoon, everyone, and welcome to our virtual seminar series, Champions for Diversity in the Neuroscience. And today we have the pleasure of having Dr. Mary Kay Lobo from the University of Maryland School of Medicine at the Department of Anatomy and Neurobiology. And Dr. Lobo, she also, in addition to be faculty at University of Maryland School of Medicine, she is co-director of the Center for Substance Use in Pregnancy and are very much involved in everything that has to do with understanding and reaching out this community and studying drug addiction and other mental illness. Dr. Lobo received her bachelor's in science in biology and a PhD in neuroscience at University of California, Los Angeles. And after that, she went to UT Southwestern in Dallas to work with Dr. Eric Nestler, who is also a leader in the field of molecular biology of neuropsychiatric disorders. And with him, when he moved to Mount Sinai, she moved to New York, and then after her years as a postdoc, she was recruited at her institution right now. A little bit about Dr. Lobo's research, which focuses on the neurobiological mechanisms that mediate maladaptive behavior, including those related to substance use disorders and mood disorders. And her lab employs a multitude of genetic and cell subtype specific tools to probe the cell type specific adaptations in the brain in disruptive behavior. And Dr. Lobo is a reviewer at the Journal of Neuroscience and eLife. And in 2017, she was the recipient of the Presidential Early Career Award for Scientists and Engineers awarded by the Obama administration. And of course, her lab research is funded by NIDA, NIMH, and the United States Israel Binational Science Foundation. So Mary Kay, thank you so much for accepting our invitation, and we look forward to your talk.
Thank you so much for that really nice introduction. And I'm really glad to be here today to talk to you all and tell you about the research in my lab, including, the impact. I'm going to focus on work where we've looked at stress and opioids and the impact on the brain mechanisms and what that might mean, for trying to develop future treatments. But before I begin, I was asked to kind of tell you about my background in science and just my background in general and how I became a scientist. So I thought I'd share with you my background. And I also come, like many of you, I come from a diverse background. Mine might be a little bit different. It's a bit unique, and I know I often, oops, I'm not going forward. I often confuse people with my identity, but one thing that's diverse about my background is I'm a child of an immigrant. My mom is from the Midwest, from Indiana area, but my dad is actually from India. And then I have a last name that doesn't sound Indian because my dad's family is from a Portuguese colony in India, hence a Portuguese, Spanish type last name. But my dad actually came here as a scientist. So I had that privilege of being a parent that was a scientist for a bit of time. And my dad actually took a boat here to get here in the New Year's Eve, 1959. He took a boat all the way from India to New York and then took a train all the way across the US from New York to California to do his PhD in geology at UCLA. He was a bit of a rebel for someone from his background. He actually dropped out of the sciences and became a journalist. He was also an opera singer and wrote poetry. So he was a bit unique from his background. But I did have the privilege of having a parent that was a scientist. So I knew that there were options of being a scientist. And I grew up in Los Angeles. So growing up in Los Angeles I had a very diverse upbringing, and that was really wonderful. And it was a place where I felt accepted with my unique identity, which was really great. And this is where I first kind of became a scientist. I'll say early on, even though I had a parent that was a scientist for a bit of time, I was always intimidated by science. In high school, I'd never imagined I'd be here today as a neuroscientist talking to you all. Science was intimidating back then, and I didn't really have the confidence to think I could be a scientist. But over time, once I started college and I started to do well, I realized, oh, this is a path I can actually take. I grew up in LA, stayed in California. I actually started out as what's called a banana slug at UC Santa Cruz for one semester. I wasn't really totally happy there. So then I came back to Los Angeles and went to a community college for a few years. And this is really where I began my path in science. I started taking biology courses. I was really interested in molecular biology. In the back of my mind, I was interested in neuroscience, but I didn't really recognize at that point that I could have a career doing both neuroscience and molecular biology. But I started, to take classes and do well. And then I transferred to UCLA. And during that summer I transferred, I was actually in a summer, what's called a summer bridge program, where I applied for this program and I got into a program and spent summer doing research. And I, in my essay, I broadly wrote, I'm interested in molecular biology, genetics, but I'm most interested in the brain. So they actually put me in a neuroscience lab, and that was my first experience in neuroscience. And I spent time as an undergrad in that lab, spent time as a technician in that lab, and then recognized I want to continue to do this, and I need to go on and do my PhD. And then I stayed at UCLA. And I was really lucky because before the lab I was in before wasn't really doing molecular biology, but they were doing more physiology or histology in the brain. But the time I started graduate school, there was a PI named William Yang who just came and started his lab, and he was really interested in studying molecular biology of disease in the brain as well as normal functioning brain. And I was able to join his lab and really find a project that fit my excitement and my interests. And I started studying these cells in the brain that I'm going to tell you about today, and started studying molecular mechanisms. And during that time, I got really interested in the brain's, reward circuit and motivation. And I was really interested in how motivation is good and it's important, but it can be disrupted. So you have disease states like depression or substance use disorder. So then once I finished at UCLA, I then, as Carmen mentioned earlier, I moved to Dallas, Texas, to Eric Nesser's lab, who's really a prime person studying molecular mechanisms in reward circuitry. And then a year later, we moved to New York, which I was really excited about because I really like New York. It's a really diverse place. And then I spent the rest of my time as a postdoc there. And then a few years later, I started my own lab in Baltimore, where I continued to study molecular mechanisms in the complex brain in disorders characterized by disrupted motivation. Before I move on, I just kind of want to mention that, I started out in this path to become a neuroscientist. But as in your role as a scientist, you have, you don't just have your role as a scientist. You have so many other roles and opportunities. Once I had my own lab, I became a principal investigator. I'm running a lab. With that, I became a mentor. And various stages of the process, I was a mentor. I was a mentor when I was a technician. I was a mentor when I was a graduate student. To undergrads, I was a mentor when I was a postdoc. I'm also a professor and a teacher. I teach some courses. I was also a graduate program director for a little bit of time, and also a reviewing editor, and then also more recently a chair of a diversity committee. And also I forgot to put up here a co-director of a center. So you might start out on a path in science, but it can lead to really many different opportunities that are fun and exciting and rewarding. And you also have opportunities to make an impact on others. So that's kind of my spiels for my career path there, and I'm happy to talk about this later. But let me just segue into what we study is motivation. You could see, to lead to my career path, I definitely had a lot of motivation, and we know that motivation is really important. It's important for just daily functions in life, like eating. You need to be motivated to survive. You need to be motivated to do well in school or seek out your career in science. You need motivation for intimate relationships. You need motivations for social interactions, for hobbies, for sports. Sorry about that. My mouse is getting a little crazy here. But then there's situations where motivation can be disrupted. You have situations like, psychiatric diseases like depression, where you have a loss of motivation, you have loss of pleasure. You also have situations where, in substance use disorders, where individuals are taking drugs, but then they stop taking drugs, then they have these negative affective states where they're feeling guilty, they're feeling a lot of pain, they have cravings that leads them to take more drugs, so they can have these negative affective states, even though they're driven and motivated to take drugs. And that, I'm really interested in, what's going on in the brain and these states where you have this disrupted motivation. And as I mentioned, disrupted motivation occurs in many disorders: substance use disorder, depression. There's a lot of comorbidity between these orders. It's important to note that these are chronic and relapsing disorders. People can be okay for a while, but then they can relapse. They can start taking drugs again. They can become depressed again. And there's also limited treatments. There's not a lot of effective treatments for substance use disorder. For depression, there are some effective treatments, but it's important to note that they don't always work in everyone, and then they can work in people for some time but stop working. So we really need to go in and really understand what's going on in the brain to come up with more effective treatments from people suffering from these disorders. These disorders also cause a huge economic burden to society, and in the worst case scenario, they can be deadly. People can die from these disorders. People can die from overdoses, from suicide. So it's really important that we're really going in and trying to understand what's going on in the brain and coming up with better treatments for people that are suffering from these disorders. So as we start to study, these disorders in the brain, it's challenging. Just studying these disorders is complex. With complex behaviors alone is challenging, but then also layering on top of that, the complexity of the brain is challenging. The brain is incredibly complex. You can have many brain regions that play a role in many types of behaviors. And within different brain regions, you have different types of neurons. You also have non-neuronal cells shown here. So it's really complex. So in my lab, we really try and take a multi-level approach to really, dissect that complexity. We try to understand molecular changes occurring in the brain that occur in specific cell subtypes. So we kind of focus on more vulnerable cell subtypes in the brain and a particular behavior. And then we try to understand how do these molecular changes lead to these cellular changes. Sorry, my mouth's going a little crazy here. And how do these, when you have disruption in specific cells in the brain, how does that lead to changes in circuit disruption? How do different brain regions communicate differently? And ultimately, how does that change behavior? And if we go back and, target these molecular changes, can restore cellular changes, can restore circuit changes and restore behavior. I'll focus mainly on these molecular and cellular changes that lead to behavioral changes. I really talk about circuit changes today. So the brain region that we really study to look at these disrupted motivational states is the reward circuit. Really, it consists of some of these areas here, including the nucleus accumbens, which is the brain region we study. That's kind of a major hub in the reward circuit. It receives dopamine. I'm sure everyone's heard of dopamine input from this ventral tegmental area. It also receives glutamatergic input from this cortical area. And also has connections, this area has connections with the amygdala, kind of an emotional fear region of the brain. It's really more complex than this. And there's now that we have really cool tools in neuroscience to dissect out circuitry, we know that there's many other brain regions involved in this process. But today I'll focus on the nucleus accumbens as part of the reward circuit. But as I mentioned, this reward circuit is really important for meeting motivation for natural rewards, but it becomes disrupted by drugs of abuse. So motivation is geared toward drug seeking. And it also becomes altered in mood disorders, which are characterized by loss of pleasure and reduced motivation, which I talked about previously. And as I mentioned, we focus on the nucleus accumbens. It's pretty much every brain region is very heterogeneous. This area is heterogeneous. There's two major neuron subtypes here that send axons to other brain regions. So they're projection neuron subtypes. And I'll tell you a bit about them on the next slide, but this is just an example of a mouse line where we've tagged these two neuron subtypes with either a red fluorescent protein or a green fluorescent protein. And if you look closely, you can see that they're pretty segregated. You don't really have any overlapping cells. You have a couple of yellow cells here, but for the most part, these two cells are segregated. And before we had these tools, so these fluorescent tools started coming out around the time I started graduate school. Before we had these tools, we didn't really have good tools to study these two cells. You couldn't really differentiate them in the brain. We didn't even have good antibodies to stain them. So we've come a long way since I started graduate school almost 20 years ago, where we just have a lot of nice tools to dissect out these different cell subtypes in the brain, including these two that I'm looking at. So these two neuron subtypes in this region called the nucleus accumbens are often called medium spiny neurons or also spiny projection neurons. people call them different things. In my lab, we just like to call them, we like to refer to them based on their enrichment of different dopamine receptor 1 or 2. But here are some other molecules that they're enriched in. And myself and others have isolated these neurons and done large gene expression profiling of them to find a number of molecules. But we just call them D1 or D2 in a sense. And these two neurons are kind of thought to have this yin-yang property where their opposite or contrary forces are actually complementary, where these D1 neurons are kind of thought to be the reward promoting neurons. If you turn on activity in these neurons, you promote reward behavior, whereas the D2 neurons kind of put a brake on the behavior. And in a normal situation, you'll have these two neurons kind of acting together, right? Because you want a situation where you're motivated to do one thing, but you kind of want to put a brake on motivation for everything else. You want to focus on that one task. So they'll both be acting together to produce normal behavioral output. But in situations where in disease state, you might have an imbalance of these two pathways. I will say this is a bit oversimplified. We have seen this when we go and test mice or animals in a lab setting, but really as we start to study the more complex brain, we realize that can depend on what stimulus you're giving to an animal. It can depend on which area of the nucleus accumbens you're looking at. And it can also depend on, sometimes these especially these D1 neurons can project to different brain regions. So it could also depend on that. So it's a bit oversimplified. But it has been, we have been, like many of us have kind of shown these opposite roles. And today's talk, I'll really focus on these D1 neurons, these kind of reward promoting neurons, and some studies where we're showing that these neurons are actually shrinking. They're dendrites here, which is a point of communication where they're receiving input from other cells, from other brain regions. They're actually shrinking, so you may not have that communication. So I'll start off by telling you about the stress work in our lab. Stress is, all of us suffer from stress, and many people can handle stress. They can be resilient to stress, or they use it even as a tool to be motivated to do something. But we can have cases where stress can really lead to disrupted motivation and manifest in a variety of disorders, including depression. Stress can precipitate depression. Stress can precipitate substance use disorder, where someone who might not taking drugs might start taking drugs again under stressful conditions, and also precipitate other psychiatric diseases like bipolar disorder or schizophrenia. In the lab, the model that we use is this, what we call a chronic social defeat stress model. It's kind of this bullying model where you have this mouse here. This is a retired CD1 breeder, and it lives in this big hamster cage. And you put your experimental mouse in here, and they're exposed to each other for 10 minutes a day. And this mouse doesn't really like to have its home intruded on. So it's not a very pleasant interaction, and it can be very stressful for both of them. And then we actually put a perforated divider here. So it's a divider with holes, and we house our experimental mouse on the other side of this perforated divider. So you have all this sensory contact in place and not the physical contact. And then you do this for 10 days. They go into a cage of a new mouse each day. And then spend 24 hours with them. And then after that 10 days, we do a variety of tests. This is just one of the tests we do, which is a social interaction test, where you have this little interaction zone and you have this little chamber here. And you first test your mouse and see how it's interacting just with that chamber alone. Then you put a novel mouse here that they've never seen before and see how are they interacting with that mouse. And here's an example. Here's our control animal. You can see the control animal is running right up. It's very rewarding to go interact with a novel social target, whereas our stress animal is going up, kind of exploring, but then they go and they run away to the corner and often spend a lot of time in the corner. So this is a model we often employ in our lab. I'll say that this model, one caveat and limitation of this model is you can only do this in males, because what will happen if you put a female in here is this mouse will just want to mate with the female. We do have some other stressors going on that are kind of a modified version of the social. I'll tell you about that today, but we are able to do this, like that modified stress in females, which is really important. It's important that we're including both sexes in our studies. So one thing we found, So one thing, my first student that was working on this project, what he found, he was looking at these neurons. He's like, I think these neurons are shrinking. I was like, let's go in and look at that. And this is one of the first images he showed me of these D1 neurons. Remember, these reward promoting neurons. Here's a control animal, here's a stress animal. And you can see their dendritic arbor is dramatically different compared to the control animal. Since this very first experiment, we've gone on and quantified this multiple times. And the way that we do this in the brain is we use transgenic mice. These are mice that express Cre and D1 expressing neurons. And then we use viruses. We use a lot of approaches with adeno-associated viruses. And then what we do is we inject these Cre-inducible viruses that I won't go through the whole process, but basically these viruses don't work if Cre is not on, but in the presence of Cre, you actually end up turning on the virus, and you can express the virus of interest in a Cre neuron. So we're able to express our virus of interest or a gene of interest in these D1 neurons. In this case, what we did is we used a virus that expressed EYFP, enhanced yellow fluorescent protein, in the D1 neurons. We inject it into the nucleus accumbens of these D1 Cre animals, and we inject small amounts that allows us to sparsely label these D1 neurons. And then we go and we reconstruct the neurons. And then what we can do what's called a shoal analysis. Where you can draw these concentric rings around these neurons and then look at their intersection on these various shoal radii. And what we found, here's a control animal, here's a social defeat stress animal, a D1 neuron. What we found, if you look at these number of intersections at these different shoal radii, you see that they're reduced in our stress and our social defeat group compared to control animals. And one thing that we're seeing quite often, if you actually correlate, if you compare your social interaction ratio compared to this area under the show profile, you see that there's a nice correlation. You actually find that animals that interact less with a novel social target actually have this reduced area under the show profile. So they have this reduced dendritic arbor. We also found that the total dendritic length and as well the number of branch points are reduced in these D1 neurons after stress. So we really were interested in also looking at the D2 neurons. Are these changed as well? Just to show you, we looked at both populations. No change in the show profile, no correlation between their social interaction score and this area under the show profile, and also no change in dendritic length or number of branch points. So this is really specific to these D1 neurons, these reward promoting neurons. So we really wanted to understand what are the molecular processes occurring in the brain and these cell subtypes that might lead to this dendritic shrinkage. And we started to kind of go in and look at molecules that play a role in giving neurons their structure. So cytoskeleton molecules. And here's just an example. You have, neurons or all cells, throughout the body have a specific structure, and that has to do with molecules that regulate actin cytoskeleton dynamics. And there's many molecules in here. There's many parts, right? There's actin, there's microtubules, there's a number of components to give the cell its structure. But there's molecules that regulate all these components, and some of these molecules are these enzymes called GTPases. And what they do, there's various enzymes, there's RhoA, RAC, or CDC42. What they do is they start to regulate other downstream molecules, and that they regulate different pathways, and ultimately this regulates actin polymerization, contraction, turnover. And we were particularly interested in this RhoA pathway because this RhoA pathway, specifically a RhoA pathway, has been shown to negatively regulate dendritic complexity. So when you have high levels of one particular RhoA molecule, you get reduced dendritic arbor, which is exactly what we saw in our stress animals in these D1 neurons. So we went in and looked at this further, and we have tools in the lab that allow us to isolate RNA from a D1 or a D2 neuron. These are animals where they're called ribotags. So we actually have the ribosomes. The ribosome is the place where mRNA is being translated into protein. So it's a place where you'll find mRNA that is likely being turned into protein. So we can tag these ribosomes in a D1 or D2 neuron, and then we just use antibodies to immunoprecipitate down these tagged ribosomes. And then what we can do is we can extract the messenger RNA from these ribosomes. So we get a pure population of messenger RNA from a D1 or a D2 neuron. And that's, when you look at messenger RNA, that tells you what genes are being turned on and expressed. In this case, it's messenger RNA that's likely being turned into protein because they're found on the ribosomes. And we went in and we looked at a few molecules, especially in this RhoA pathway. We looked at molecules that were upstream, including one that positively regulates RhoA and one that negatively regulates RhoA. So it's RGAF1 and rotican. And we looked at a molecule that's downstream. And as I mentioned before, this pathway in particular, when you see high levels of this pathway, you see this negative regulation of dendritic complexity, which is what we were seeing. And when Meg Fox, a graduate or a postdoc in the lab, went in and looked at these different molecules, what she found, she looked at the D1 and D2 neurons, she found in the D1 neurons an increase of RhoA in the stress condition, an increase of this variant of ROC, this is a downstream molecule RhoA in the D1 neurons, no change in the D2 neurons in both cases. She found that this negative regulator of RhoA, this upstream regulator, is down in the D1 neurons, no change in the D2 neurons. And then this positive regulator, RGAF1, is increased. So we see this positive regulation of this RhoA pathway, which is involved in negative regulation dendritic complexity. And that fit really nicely with what we're seeing with these neurons shrinking. So Meg then went in and looked at these, she went in and then decided to target RhoA in these neurons. We use viruses, adeno-associated viruses. She made what's called a dominant negative RhoA. So this is kind of a variant of RhoA, but what it does is it outcompetes the regular RhoA. So the effect it has is to block RhoA, the active form of RhoA. And then we've validated that it is working that way. I'm not going to show all the data. And again, she put it in this virus that's only expressed in Cre. It turns on in Cre expressing cells. So we can only express it in these D1 neurons in the nucleus accumbens. And here's just some validation, some like beautiful fluorescent images showing that our virus, which has the EYFP tag, is expressed there. Because this is a variant of RhoA, you can just use an antibody and stain for RhoA, and you see that there, and you see a nice co-localization between the two. So what she found, what she found is, let me go going a little bit crazy here. My mouse gets a little bit crazy. Sorry about that. Okay, so what she did is she did the virus surgery. She waited. You have to wait a few weeks for the virus to express. She waited till that dominant negative RhoA was expressed in these D1 neurons, and then she put animals through this full social defeat stress paradigm, and then did the social interaction test, and then did some downstream analysis looking at the neuronal morphology. What she found, if you look at the social interaction ratio, here's our control animal. There's really no difference between the animals that received EYFP in the D1 neurons or dominant negative RhoA in the D1 neurons. However, when you look at the stressed animals, you see our EYFP group, which is a stress control group. You see that they have this reduced social interaction, which I showed you previously. And here is an example. You can look at a heat plot of this. You can see that these animals, we have these darker colors. It's where the animals spend more time. And you see that the EYFP group and the stressed animals, they're spending more time in the corner. But the animals where we actually block that RhoA activity with this dominant negative RhoA, we actually have an increase in the social interaction compared to the control conditions. So you could see that the animals will spend much more time here. And just to point out, just giving, dominant negative RhoA or EYP alone in control conditions doesn't affect, these are control animals that weren't stressed. It doesn't in fact affect their social interaction. Meg then went and looked at the neuronal morphology, and here's an EYFP. This is both, these are examples from stressed animals. Here's EYFP, here's dominant negative RhoA. And you can see here that you see the EYFP stressed neuron, the D1 neuron. You see this dendritic arbor, but then you can see that you can enhance it when we block that RhoA activity. So not only does blocking that RhoA activity and these D1 neurons improve behavior, it also enhances this dendritic arbor. So it enhances the dendritic complexity. So this is great. You know, we can go in and target neurons in the brain and, you know, for specific molecules and then change behavior, change cellular properties. But the reality is in humans that are stressed and might be depressed, we're not going to be able to do that. We're not going to express a virus in a specific cell subtype. We don't have transgenic, you know, people that express Cre. But what we can do is get information about these molecules and try to think about pharmacotherapies that might be related to what we're doing here with these viral approaches. So I mentioned before, you know, I showed you before this RhoA pathway, and I mentioned this molecule ROC, which is downstream of RhoA. I showed you that this ROC2 variant is increased in these D1 neurons, the stressed animals, no change in the D2 neurons. And then here we actually found, if you looked at activity, like you can look at the active form of ROC, which is going to activate these downstream molecules and activate these actin properties, you see that it's increased in the stressed animals. So it turns out there's actually a lot of ROC inhibitors available. So what my lab did, and Meg specifically, she went in and tested a bunch of ROC inhibitors. But we kind of took a different approach because we thought, okay, this is, you know, we want to kind of mimic this like, you know, the human condition, right? People don't go to the doctor, you know, before they're stressed, before they see, you know, before they display, you know, psychiatric disorders. They go when they're actually sick. So someone that's depressed is going to go when they're sick after they've gone through a stressful period or other situations that lead them to develop these, you know, lead them to develop behaviors like anhedonia or reduced motivation. So we decided to put animals through the full social defeat stress. She also labeled the neurons with that EYFP, so she can look at the neurons afterwards, put them through the whole social defeat stress, did the social interaction, and then she gave them this, what we call a therapeutic treatment of this ROC inhibitor just over a week, and then did the social interactions, did another test. I'm not going to show you this test, but she's done some other tests downstream, and then collected the tissue. And what she found, the animals, so she did a social interaction here and then another social interaction. And you can see here, here's our control animals, really no difference, you know, pre or the first social interaction, pre-treatment, second social interaction, post-treatment in both our vehicle and our ROC inhibitor condition. When you look at the social defeat stress group, you see the vehicle, the animals that receive vehicle have this low social interaction score. And here's pre-treatment. They're not really spending much time interacting here, and post-treatment, because we've just given them a vehicle, they still look the same. They're not really interacting with a novel social target. You see that the animals that receive the ROC inhibitor, so this is pre-treatment, they have this low social interaction score. They're spending more time in the corner, as shown here. The post-treatment, some of them are showing this improvement in behavior where they look similar to controls. And if you just look at these, you see for the most part, they're all improving. Some of them don't improve to control levels, but they started out much lower. But for the most part, overall, you're seeing this improvement in the social interaction score. And here's an example of an animal that's spending more time in this social interaction area when we've given this ROC inhibitor treatment. She also went in and looked at the neuronal morphology, and you can see here, I mean, just by looking at these neurons, you do see this, you know, very expansive, you know, these expanded dendrites here. Oops. Sorry, my mouse likes to go crazy when I do these presentations. So you see this expanded dendritic arbor here, and then here it's quantified where the animals that receive, these are the stressed animals, that receive this ROC inhibitor, we're improving this dendritic arbor here. So we're able to kind of use this ROC inhibitor as a therapeutic treatment in our stressed animals to improve behavior and improve this dendritic arbor. And this is something we can think about in the future. You know, is this a viable candidate for you know, potentially, you know, them treating depression? So now I kind of want to just switch gears in the last few minutes and talk about our interest in looking at opioid use vulnerability and stress vulnerability, because these two behaviors can kind of interact. You can have situations where someone is stressed and that leads to increased opioid use vulnerability, but you can have situations where individuals are taking opioids, they're using these substances, and they might be more vulnerable to stress. And that can kind of lead to a cycle of like stress that leads to drug taking. Drug taking can lead to stress vulnerability. And one thing that we were really interested in is a previous study from over 20 years ago actually looked at these medium spiny neurons after abstinence from an opioid morphine. And this is just, back then we didn't have tools to look at D1 or D2 neurons. This is just looking at all the medium spiny neurons. And here's a control neuron. This is a neuron from animal who received morphine and after abstinent time period when they're not taking morphine for a while. And here you can see then when they look at branches and these medium spiny neurons, they're reduced in the morphine group. When you look at ring intersections, so doing the RhoA analysis, that's reduced. And you can also see like there's like I didn't talk about dendritic spines, but these are also points of contact where you have synaptic interactions with neurons that send inputs. We see that these spines are also reduced. So we were particularly interested because there's something about this abstinent time point. If you look at this like cycle of drug taking, you have individuals that are binging on drugs, then they stop using drugs and they go through this very immediate withdrawal state, which we have these more physical withdrawal symptoms. But then if you wait longer time periods and you wait for these more prolonged abstinent time periods, you can often see negative affective states, especially with opioids. You see these negative behaviors like anhedonia or loss of motivation. And it's that kind of negative state that leads to, thinking about the drug more and craving, and then ultimately leads an individual to taking the drugs again. So we're really interested in this abstinent state and looking at this time period where they're thought to be negative affective states. And we're specifically interested because you see these changes that are very similar to stress, where these neurons are shrinking, at least when they look at all these medium spiny neurons. We wanted to go in, and now that we have the tools, we can look at these different medium spiny neuron subtypes. So we were really interested to see, are there similar dendritic adaptations in the D1 MSNs that we saw in stress susceptible conditions? Are these also occurring in these opioid abstinent conditions? And we wanted to focus, we were trying to decide what opioid we should focus on. And here's just a chart of the national drug overdose deaths. And I'm sure everyone knows that there is a major opioid epidemic going on. And one of the things, you see a number of prescription opioids have increased over the years, as well as heroin. But this yellow line here is really a major contribute to this opioid epidemic and people dying from opioid use. And that's the use of synthetic narcotics, including fentanyl. So we decided going forward with experiments that we would focus on fentanyl. Here's another example of the different types of opioids that have been used over the decades. In the 90s, there was a lot of prescription opioid use. Early 2010, there was heroin use, and then more recently, since 2013, synthetic opioids like fentanyl. And one reason people are actually dying is because fentanyl is much more potent. So it's actually 15 times more potent than heroin. So you just need this amount of fentanyl to get the same effect as you would with this amount of heroin. And the situation was so bad that even here in Baltimore, they were like handing out signs and having flyers showing, telling people like, be careful, don't let fentanyl kill you, because overdose is real. A lot of people were dying from fentanyl on the streets, so they're sending a message, please, like, be careful in using fentanyl because it can be deadly. So when Meg started these projects, what she did is she just gave animals, both males and females, fentanyl in their home cage over 5 days. That seemed to be enough time to see effects. And she just wanted to make sure, is there anything, are they consuming the same amount of fentanyl as water? Because we have a water group as well, and there's no difference in their overall liquid consumption. She also wanted to make sure that this 5 days was enough to really have an effect. So I mentioned you get these early physiological withdrawal states after you stop taking opioids. And she looked at a number of behaviors to look at this withdrawal, and she found that her fentanyl group, and this could be things like just like teeth chattering. You have these things called like wet dog shakes and other types of behaviors. And she found that the fentanyl group had a higher withdrawal score. So that's reassuring because it shows that the fentanyl is having an effect in these animals. So she then went in and did that 5 days of fentanyl in the home cage, or the control group got water. And then she gave everyone water for 10 days. So that's that abstinent period. And then what she first did was just did a social stressor. This is just a 1-day social stressor. The one I showed you before, this chronic social defeat stress, was 10 days. This is just a 1-day stressor where you get 3 stressors with this CD1 retired beater in 1 day, and the next day you do a social interaction test. Like our other behavior, we can only do this in males. Like I said, we've worked out a female stressor. We haven't really worked out this 1-day stressor in females. So this experiment is males, but I'll show you a few experiments that we did looking at behavior in females after this. So after this 1-day stressor, this isn't really enough to cause a social avoidance phenotype that I showed you before with the 10-day stressor, unless the animal is more vulnerable to stress. So what we found out when Meg ran these animals, and here's the graph quantifying it. If you look at water fentanyl groups that didn't undergo stress, they're interacting about the same. However, when you look at the stress group, you can see that the groups that got fentanyl have this reduced social interaction compared to the water stress group, compare that 1-day water stress group. And here's just a heat map showing the animal that received water in 1 day of stress. They're spending more time interacting with a novel social target, whereas the animal that received fentanyl will spend less time and often more time in the corners. She also wanted to look at other behaviors. One behavior she did is, similar to the other behavior, but it's a 3-chamber social preference test. You can do this in both males and females. And the way this works is you have 3 chambers and 2 cups. One is an empty cup here. One you have a same-sex conspecific. And here's an example. This is actually a non-stressed and a stressed animal. And you can see it's hard to see, but there's a same-sex conspecific in these top cups. And you can see in this animal here is interacting quite a bit. It's a novel mouse they've never seen before. It's rewarding. Where this animal here is actually not spending as much time in avoiding this animal. And this, as I said, this is an example where we've actually done stress in females. This is a non-stressed and a stressed female. But we just went in and employed that, just looked at social preference without stress in our fentanyl and water groups. And again, we did the same paradigm, but this time we used both males and females, did the 5 days of fentanyl and the 10 days in the home cage, and then the social preference. And you can see here, when you compare the water to the fentanyl group, you see overall there is a significant decrease in the social preference. We are seeing a range of behaviors, and it looks like maybe there's individual differences, and that there's some animals that are interacting fine compared to controls, but there's some that have this reduced social interaction. It seems to be happening more in the female group shown here. And here's just a heat plot. Again, you see an example of a fentanyl animal that's not spending much time with this novel social target compared to the water animal that is. Finally, Meg did another behavior, which is, sorry, my mouse again is going crazy. Okay, my mouse. Okay, well, I won't touch anything. Okay, so she also did this behavior. It's an elevated plus maze, and I don't know if anyone's familiar with it, but basically it's this elevated maze, and you have 2 arms. You have a closed arm that's kind of a safe space because the mouse is really high up, so it feels safer in this enclosed area. Then you have an open arm. It's kind of like walking a tightrope here. She put both males and females through that fentanyl paradigm abstinence and then test them on the elevated plus maze. And here it's quantified here, where you see animals will spend less time. The fentanyl animals will spend less time in this open arm here, so they're not exploring this kind of novel, more dangerous environment, and more time in the closed arm, where it's a little safer. And here here's a heat plot. You can see the water animals really spending similar times in both regions, whereas the fentanyl animal will tend to spend more time in this closed area where it's safer. So they have this kind of enhanced avoidance behavior, or this, reduction exploratory behavior. So, right, we saw all these changes that were reminiscent of stress in these behavioral changes, reminiscent of stress in the fentanyl group. So we want to go in and see, are we also seeing these changes in dendritic complexity in this fentanyl group in the D1 neurons? So she did the same experiment. She labeled the neurons, put them through 5 days of fentanyl and 10 days of abstinence, and then did a shoal analysis on these neurons. And basically what she found when she looked at the D1 neurons, you see in the fentanyl group, this fentanyl abstinent group, you see this reduced show profile, you see reduced dendritic length and reduced branch points. And here's an example of a fentanyl D1 neuron compared to a water neuron, where you see this dendritic atrophy and no change. Looking at all these various analysis in the D2 neurons, no change. So in these D1 neurons, we're seeing the same effects as stress. We're seeing that opioid abstinence leads to similar changes in behavior and similar changes in dendritic complexity in the D1 neurons. So finally, just in the last couple slides, I want to show you that we want to go in and try and understand what's going on a molecular level in these neurons that might be leading to these changes. We can go in and manipulate those genes and maybe lead us to potential pharmacotherapies. We use that same ribotag approach where we isolate D1 or D2 neurons, put the animals through the fentanyl paradigm. We had to pool a lot of animals instead of being a lot of animals. We used both sexes. And what we found is that we had a lot of differentially enriched genes. When you compare fentanyl to water, we have more differentially enriched genes in the males compared to the female, in the D1 neurons compared to the D2 neurons, and only 4 common overlapping genes. We ran both sexes. We don't really see many sex-specific changes. The other thing we've done with this data, we could take these data and look at these large network changes. And we did this analysis that's called weighted gene co-expression network analysis. Don't worry if you don't understand that. The point of this analysis is actually lets you group genes into what we call our modules. So you can group these large genes into one module where they're represented by similar gene expression patterns. You actually have genes changing in a similar way. So we get all these modules, and it's kind of like, what does a module mean? It just means a bunch of genes are connected, and they have some genes that are more highly connected called hubs. It's kind of like when you think about flight paths in across the world or the US, where you can have all these different cities are connected by their flights. But then you have some hubs. This is Delta. There might be a hub in Detroit or some other regions, which are these genes that are kind of like the main hubs for these modules. Don't worry if you don't understand that. Even I have trouble understanding it, but it's a way for us to break the genes up into groups and look for patterns in those groups. And this is just a plot showing different modules. We just color code the different modules. And then here, this is the outer circle of different modules. The inner circle is a D1 versus D2, just the change in the gene expression. And this last circle is just the P value. And what you can see here, this is the D1 circle. You can see there's more color here, which basically just means there's more modules that are changed, that are significantly changed by fentanyl in the D1 neuron. So there's 9 D1 neuron modules and only 2 D2 neuron modules. And the thing I want to point out is in these modules, while they might not always have biological similarities, the genes within these modules, we were able to find biological similarities. And what Meg found is that a number of these modules, these D1 modules, actually contain many dendritic morphology genes that are downregulated. And that makes sense because we see these D1 neurons shrinking. And specifically, she found this green module where she found a lot of dendritic morphology or even plasticity molecules that were downregulated. So we're now trying to go in and focus on specific molecular pathways in this green module. We can then make viruses and use our transgenic mice to target the molecules to see if if we target these molecules, can we improve behavior and improve this dendritic complexity? And then we can hopefully use this information to give insight into potential pharmacotherapies. So just to summarize, I've shown you that both stress susceptibility or opioid abstinence leads to dendritic atrophy in these D1 neurons. Through stress, we found it's through this RhoA pathway. Fentanyl exposure and abstinence. We found that these animals that were abstinent from fentanyl have these leads to changes in stress or negative affective states. We found that there are gene expression patterns after abstinence from fentanyl, and we seem to see more of them happening in the D1 neurons. And then we found a lot of dendritic morphology molecules are reduced in the D1 neurons in the fentanyl abstinent conditions. So just to summarize, I've shown you how we can find changes in molecules that lead to cellular changes. I didn't really tell you much about circuit changes, but ultimately this leads to changes in behavior. And what we can do from these molecular changes is hopefully down the road, find molecules that could potentially be targeted with pharmacotherapies for treatment for disorders that disrupt motivation. So I just want to thank the people in my lab. Particularly, a lot of this work was driven by Meg Fox, a postdoc in the lab, and thank my various sources of funding, as well as everyone else in the lab. And this is how we interact these days during COVID times. And I'm happy to answer any questions. I think I went a little bit over, especially 15 minutes for questions, so sorry about that, but happy to answer any questions.
Thank you, Mary Kay. That was a wonderful talk, very interesting as well. We have a question from Jose Garcia, and he says, how was the RhoA inhibitor administered? Is there any secondary effect?
So the experiment I showed you, we used a ROC inhibitor, and that one we gave systemically. We have actually given RhoA inhibitors, and we've given them both systemically and into the nucleus accumbens, and we've seen improvements in the stress behaviors when we've given them in both conditions. We haven't done the ROC inhibitor into the nucleus accumbens, but the experiment I showed you was just injecting systemically.
Okay. And there's another question, says, thinking about extending this to humans, I could imagine some people going through fentanyl withdrawal might also be experiencing additional other stressors. So have you looked at combining your fentanyl paradigm with additional stress paradigms to look for additive effects?
Yeah, and we haven't done them both at the same time, but we have done experiments where we've stressed the animal beforehand. We've put them through the full social defeat stress, or as I mentioned, I didn't show you, we use this, what's called a witness defeat stress in the females. And then we've looked at their fentanyl intake, and we're actually seeing sex differences where the females are actually taking more fentanyl, at least in this where the animal's undergoing this witness stress paradigm. So we are looking at ways where we can give stress before the fentanyl afterwards and vice versa, but we haven't done an experiment. That's a good thing to do, experiment where they're going through stress and fentanyl at the same time, or fentanyl abstinence, and those are things that we're thinking about. We just haven't done yet.
Okay. Then we have another, one of our students says, what about the pain stress that can be the initiation of the stress and opioid usage cycle? Can that be taken into account in that study?
Oh, the pain, like a pain factor in the stressor. So right, like this actual social defeat stress, there is a physical stressor there, and that could potentially cause wounding and pain. That's why when we're trying to use these witness defeat stress paradigms, when we look at opioid intake after stress, so that in those witness defeat where animals just watching the defeat episode, and we have data, and other people have published, our colleague Sergio Dominguez has published on this, Carlos Bolanos' lab has published on this, where they see these animals witnessing this stressor actually show these stress susceptible phenotypes. So for conditions where we want to look at opioid intake after stress, we're focusing on these witness models where there's no physical interaction, there's no pain interaction.
Okay. And I had a question, Mary Kay, with regards, because you see a very robust difference between the D1 medium spiny motor neurons and the D2. And how is the distribution of the mu and kappa receptors in these neurons? Are they differentially also, which will go toward the effects on the fentanyl? Could you elaborate?
Yeah. So I don't think the mu has been, I mean, I know, so I don't think I don't know that anyone showed any differences at the level of the receptors. There is like the D1 neurons do express dynorphin, whereas the D2 neurons express enkephalin. So they express different endogenous opioids, and that might play a role in these responses. But I don't know. And people have looked at conditions where they've, manipulated at least mu. I don't know if they've manipulated kappa in these two neurons. But I don't know that there is differential expression. People always ask us, are these receptors changed after the opioids? And we're not really seeing a difference there. But there could be something at the level of, regulation of these receptors, which we haven't looked at further in the opioid abstinence. Or the endogenous opioid system, which is different in these two neurons. So I think it's something that hasn't been well addressed yet.
And so I don't know if I don't remember you mentioned about the core and the shell. So
Yeah.
Do you see? Are you looking at the shell? Or are you looking?
Yeah. So I don't know. In mice, people are getting better at targeting core and shell. So we haven't done so much targeting, and it's it's hard. Well, one thing, it's hard to dissect core versus shell for these molecular experiments. But one thing we're starting to do now a lot of is RNA scope, where you can do in situ hybridization, and there we'll be able to look at our gene changes and see is there a change in the core versus shell. I'll say that Meg in the lab, she is following up on some targets that she found from this gene expression data, and she is using RNA scope and looking at those targets in the core versus shell. And there might be more, more change in the shell, but we're still kind of trying to figure that out. And there's also different regions in the shell, and Michael Brukus's lab has shown, and Reem Al-Hassani, a previous postdoc in his lab, has shown, depending on where you stimulate those D1 neurons in the shell, you can have an aversive response or a rewarding response. So it's really important that we're now going in and looking at these different populations to see if is this dendritic atrophy occurring in just one subset of D1 neurons or across D1 neurons in core versus shell. So that's something we want to do, and I think we can't do that at the level of the large scale gene expression because it's just hard to dissect out core versus shell. My postdoc lab was able to do that in rats, where they could punch the core and then punch the shell afterwards. But in mice, it's hard. But we can use it by just using RNA scope and using the sparse labeling is tricky because it's we're just limited to what we can label. So we kind of just group them all together, but we need to be better about going in and looking at the different regions. So it's something we're thinking about. We just haven't gotten to that point yet.
Okay, and then the D2 group that it resonates to me with psychotic disorders or things that are beyond the maladaptive disorders that you're talking about. Do they have some correlation with that? Are we looking at targets for these kinds of disorders through these D2 medium spiny neurons?
Yeah, I think, it's funny. It's a lot of people just seem to focus on the D1 neurons, and maybe there's just more obvious changes in the D1 neurons, but I think it's important to look at the D2 neurons. We are seeing, I didn't show you this, but after stress, what we see in the D2 neurons is there is an increase in dendritic spines in these neurons. We've published that, and at least some preliminary data with the opioid abstinence, we're seeing the same thing. So I think we need to look at those, we need to be looking at those populations as far as antipsychotics. I don't know if people, have really, and we certainly haven't looked at that as antipsychotics in these paradigms, and that would be interesting to see, how antipsychotics are influencing these D2 cells, and are they, can they, we just don't know if the antipsychotics can change the behavior and change the cellular changes. And we, the answer is we need to be looking more at the D2 neurons, not just to focus on D1 neurons. And people are starting to do it. It's just it's easier to focus on the D1s because there's so many, these really obvious changes, but we're definitely interested in looking in the D2 neurons.
That sounds I'm very interested in that stuff. Glad that you share my ideas.
Okay.
Well, I
Great.
think we have reached our 6 o'clock time, and then we really appreciate you being here. And we thank everyone to coming, and please come next time, because next time we're going to talk about a very different topic, which is neuroscience and sociology. So we're going to see a topic about the taboo of race, colorblind racism, and the violent brain. So it's a very interesting topic, and I hope everyone can come and be at this talk with Oliver Rollins. So thank you, Mary Kay, so
Yeah,
much
thank you.
for being here.
Yeah.
And I look forward to seeing you again sometime.
Yeah, I hope to see you in person at some
Yes,
point in the
we
future.
will. At least in SFN somewhere. We'll find
Yeah.
it. I'll
Yeah.
make sure of it. Thank you, Mary.
Okay. Okay. Thank you. Take care, everyone.